genes densities? What is gene density? How many genes are there per unit length of the DNA is called as gene density. The example can be some of the genomes, they have less gene densities, and some of the genomes, they have high gene densities. In case of mitochondrial genomes, they approximately have 20 genes in their entire genome. While on the other hand, free-living bacteria and archaea have 1,000 to 7,500 genes. In case of fungi, unicellular fungi, they have approximately 5,000 genes. In case of multicellular organisms, they have genes somewhere between 20,000 up to 40,000. 1 million base pair length in human genome have approximately 10 genes. So this is example in case of human genomes that in 1 million base pair, there can be up to 10 genes. 1 million base pair of bacterial DNA contains 500 up to 1,000 genes. So this is the comparison between a prokaryotic genome and a eukaryotic genome. In case of eukaryotic genome, there can be 10 genes in 1 million base pair, while in case of prokaryotes, in case of bacteria, in 1 million base pair, there can be 500 up to 1,000 genes. More gene density in case of prokaryotes, as we have seen that there are more gene density in case of prokaryotes, more genes per base pair. So bacterial genes, why this density is more in case of prokaryotes? There are different reasons that in case of prokaryotes, they have a high gene density. Number one, the bacterial genes have no introns. Second, there is less intergenic DNA, mean the DNA sequence between two genes is less as compared to eukaryotes. Although in some of the prokaryotes, we do not see any intergenic DNA. In case of prokaryotes, other reasons can be neighboring genes, they are very close to each other throughout the genome. The fourth can be hardly we found any big region of non-coding DNA in case of prokaryotes. So these are some of the reasons that why prokaryote they have high gene density as compared to eukaryotic genomes. Although the gene number is not correlated to the genome size, the example of that the gene numbers are not correlated with the genome size can be of the C. elegans and Drosophila because both they are eukaryotes. So in case of C. elegans, which is also called as nematode, that has 100 MB of DNA and there are 20,000 genes in case of C. elegans. While on the other hand, Drosophila, the genome size of the Drosophila is 165 MB, while there are 13,700 genes on the genome. So here we can see that the gene number is not directly related to the size of the genomes. Vertebrate genome, more peptide due to alternative splicing. Although we have seen that in case of vertebrates, the gene density is less, but there is a process, there is a phenomena which is called as alternative splicing. Due to alternative splicing, one gene can produce more than one proteins. So, although the number of genes in case of vertebrate genomes is less, but these genes, they can produce more than one proteins due to the process of alternative splicing. Humans and other mammals have low gene density or the number of genes in a given length of the DNA. Eukaryotes, many introns are found in case of eukaryotic genomes. There are introns within the exons as well as non-coding DNA is found in case of eukaryotes. This is a table that describes the average gene densities in case of different species. Here we can see that in case of humans, size of the genome, estimated number of the genes, and average gene density. So here we can see that on average, there is one gene in 130,000 nucleotides. In case of mouse, the size of the genome, there are approximately 22,000 genes on the genome of the mouse, and one gene can be approximately in 113,000 of the nucleotides. Drosophila, in case of Drosophila, the estimated genome size, estimated number of genes, somewhere between 13,600 up to 13,700, and the average density of the genes. Average gene density in some other species, like in case of Arabidopsis, in case of C. elegans, in case of Cerevisiae or yeast, or in case of E. coli. In case of Arabidopsis, the genome size, number of genes, and one gene per 4,000 nucleotides. In case of C. elegans, genome size, the number of genes approximately 19,100, and one gene is present per 5,000 base pair. In case of yeast, genome size, number of genes, and gene density, one gene in 2,000 nucleotides. In case of E. coli, we can see that the size of the genome, number of the genes, and gene density. So here we can see that in case of E. coli, one gene is present approximately on 1,100 nucleotides. While on the other hand, in case of human genome, we have seen that one gene is present in approximately 130,000 of the nucleotides. So this is the comparison between the gene densities in case of eukaryotes and in case of prokaryotes. If we conclude that what are gene densities, that is the number of genes which are present in per length of the DNA. Gene densities are usually large in case of prokaryotes, and the gene densities are usually less in case of eukaryotes. There are different reasons why gene densities are more in case of prokaryotes. Some of the reasons they may be that there is no intergenic DNA, there is no introns, and the genes they are placed side by side in case of prokaryotes. While in case of eukaryotic genomes, the genome size is large, the number of genes they are less, although one gene in case of eukaryotic genomes, one gene can produce more than one polypeptides. It means the number of proteins which are produced, they are larger when we compare it with the number of genes which are present on the eukaryotic genomes.